Okay, everyone. I think everything's working on YouTube. This morning, I'd like to say good morning, hello to everyone who's here today. Good morning if you're in the U.S. Good afternoon if you're in Europe and elsewhere east of us. My name is Lauren Hayes, and today I'm the host of today's fine seminar, and I'm glad that everyone's here today. Just a couple of quick things. At first, I want to thank last week's speaker, was Marcus Zoldo, who gave us a really interesting talk on the social lives of African mole rats. Next week, we have I think one of our rising stars, I think is hosted by Karsten, if I remember correctly. Erica Vanderwall will be speaking to us about vervet monkeys. Today's seminar, just a reminder, it's about a 15-minute seminar, after which we have plenty of time for discussion. If you're on YouTube, please post your questions in the chat, and one of us will relay the questions to me. There will be a bit of a delay. It can be up to several minutes sometimes. So please just be patient, and we'd love to hear your questions coming from YouTube. If you're here live today, as always, we post question marks in the chat. And then just put the question mark symbol in the chat, and I'll call on people as they come in. And as always, we really like to encourage our junior folks, the students, postdocs, to ask questions. We love to have them participate in this fine. So this brings me to today's speaker. I'm very happy to introduce Professor Karen Kapheim. Dr. Kapheim is a professor at Utah State University in the United States. She has a really interesting training, a fascinating training. She started out working with vertebrates. She was in Kay Holikamp's lab as an undergrad, and apparently we were across the hall from each other when I was doing my master's, and she was working as an undergrad. Didn't know it. Karen then, after working in Kay's lab, spent some time in Kenya to work with the hyenas. And then she moved on to do her PhD at UCLA. I believe it was Peter Nonok's lab. And then did a postdoc with Gene Robinson at the University of Illinois, which at some point she realized that working with vertebrates was a real challenge and decided to work with invertebrates. And there are plenty of reasons to do that, I suppose. And her research has addressed a really fascinating avenue of research. She works on, and her website describes her research program as dealing with evolutionary processes involved in plasticity of complex traits. And in particular, she's focusing on the social behavior, the evolution of social behavior in bees. She uses both developmental and sociogenomic mechanisms, studying how these underlie behavior, and therefore helps us to understand how these behaviors evolve. And her work is truly integrative. As you'll see today, I imagine, her work delves into evolutionary biology, behavioral ecology, but also uses comparative genomics, transcriptomics, physiology, neuroscience, metagenomics. So quite an impressive display of disciplines that her work influences. And I think we'll be seeing that today. Not surprisingly, she's published very well. She has over approximately 50 papers in peer-reviewed journals, great journals like Tree, Biology Letters, Scientific Reports, Prox B. And there's a science paper on that seemed like it was a really important one from 2015, looking at genomic signatures of evolutionary transitions from solitary to group living strategies. She just told me she has just learned recently or will be getting a big NSF grant to support her work starting in May. So it's a big kudos to her. And I was really impressed too by looking at her website of sort of the range of people she's training in her lab. She works not only with PhD students and postdocs, but has master students, undergrads. And I imagine that, as in addition to being enjoying, that's quite enjoyable, that's quite challenging. You're dealing with students at all different levels, and that takes someone who's got, a good view on science, I think, to do a good job at that. And so I'll end there. And today she's going to be presenting to us a talk called entitled Causes and Consequences of Behavioral Plasticity in Bees. And with that, I'll turn it over to Karen. And thank you, Karen, for being here with us today.
Thank you. Okay, let's make sure I get this screen sharing situation right. Does this look good to everybody? Okay, great. Okay, so my screen's over here, so I'm going to try to make sure I'm looking over here. Well, yeah, so thanks so much for inviting me to be a part of this seminar series. I've watched so many of these online, and many of the people who have presented their research before me and as part of this series have been really influential in my own approach to research. So it's really quite an honor to participate on this side of things. So today I'll be telling you some of what my collaborators and students and I have been doing to try to understand the causes and consequences of behavioral plasticity in bees. And so I just want to start way out here with this seminal work, The Major Transitions in Evolution, published nearly 30 years ago now by John Maynard Smith and Eros Sidmari. And in this publication, they listed, or they identified a list of the major transitions in evolution, many of which could be characterized by a transition from reproduction at the level of the individual to reproduction at some higher level of biological organization. And they point out, given this common feature of the major transitions, there's a common question we can ask of them. Why did natural selection acting on entities at the lower level not disrupt integration at the higher level? And so in other words, losing the ability to replicate independently is associated with fitness costs to individual replicators. And so one can ask then, how can this evolve through natural selection? And this has really been the question that's driving my research since starting my career as a graduate student. And I've focused on one of these major transitions in particular, and that is the origins of eusociality, where several species of insects have evolved a social system that's so highly coordinated that they function as if they're one superorganism made up of many thousands of individuals. And so the question that I've been trying to answer is, how do these superorganisms that replicate at the colony level, such as these honeybees here, evolve from independent replicators or solitary bees, such as this alfalfa leafcutter bee here? And in bees, this evolutionary transition has occurred many times in several ways, and there's a ton of variation in the social lifestyles of bees. It's actually really hard to put them into categories. But for the purposes of this talk, I just want to get us all on the same page. So I'm just going to give you a couple different definitions that help us to think about categories of social lifestyles in bees. First, there have been several independent origins of sociality, from a solitary lifestyle to the most basic forms of eusociality. These are characterized by overlapping generations, so mothers and daughters coexisting in a nest together as adults. When they do this, they tend to form a reproductive division of labor, where the mothers become the queens who specialize in the egg laying, and the daughters become the workers who remain either functionally or maybe physiologically or morphologically sterile. And then they engage in cooperative brood care, where the workers are not laying eggs of their own, but instead they're helping to raise their siblings. Now in bees, we've also had a few independent elaborations of eusociality, from its most basic form to the more complex forms that really resemble those superorganisms. And with this, we get colony level reproduction. So like a honeybee queen can't go out and start her own nest. She needs to bring some workers with her. We get specialized caste either in morphology or behavior or physiology. And these tend to be really large colonies, numbering in the tens of thousands of individual workers. And of course, with that, we need sophisticated modes of communication. And so the framework with which I've been studying these transitions is within this framework developed by Nico Tinbergen about 50 years ago, which asked these 4 questions about animal behavior, or really any trait. And that is, how does it work? How does it develop? What is it for? And how does it evolve? And these have since Tinbergen been sort of split into these dyadic relationships, like proximate and ultimate levels of analysis, as coined by Ernst Mayr. We can also think of them as 2 different timescales, looking at things in terms of current function and mechanisms and historical timescales. But what's really important here is that Tinbergen himself stressed in the original publication where he presented these 4 questions, really stressed that they shouldn't be asked in isolation. We should really be using the answers from one of these questions to inform the others and integrating across them. And we've been doing a really good job of this in the past 10 to 20 years, synthesizing between mechanisms and adaptive functions, especially with the sort of rise of sociogenomics and our ability to look inside the genomes and understand how that translates to fitness at a population level. But we can also learn a lot by integrating across timescales with levels within a level of analysis for both proximate and ultimate types of questions. And then we can certainly do more to synthesize across proximate and ultimate levels of analysis at additional timescales, looking into the past, asking about the role of development. And so when we start integrating across all of Tinbergen's 4 questions, we can identify additional questions that can maybe lead to further insights about behavior. And this, at least this is sort of what I've proposed in this recent review paper from a couple years ago. But this here is going to be the framework for my talk today, as I share some of the insights that my students and collaborators and I have gained from over 10 years now of studying the social lives of bees. And so I just want to sort of put a little disclaimer out there. This is a lot of questions to answer in a 50-minute seminar. And so I'm just going to be giving you little snapshots, little vignettes of results, with sort of the understanding that there are a lot of gaps, there are a lot of caveats, still many outstanding questions, and I'd be happy to discuss any of those during the Q&A portion of this talk. Okay, so let me introduce you to the bees. Here I'm showing you the evolutionary relationship of some of the species that I've studied in some fashion, maybe just looking at their genomes, maybe just looking at their behavior, some combination of both. You can see I've color-coded the names of these species according to how social they are, with blue as solitary and red as representing those superorganisms. And I've starred here a couple independent origins of eusociality, one in the apids and one in the, or at least a couple in the Hellictids. And so here I just want to zoom in on this group here, the Hellictids. This is the family Hellictidae. They're otherwise known as the sweat bees. There are several different subfamilies of sweat bees, and they're all ancestrally solitary. So came from solitary ancestors and have remained solitary through evolutionary time. But if we look here at this subfamily Hellictinae, we have at least 2, but probably many more independent origins of eusociality in this group. And so if we zoom in here on the ogochlorines, we find Megaloptogenellus, and this is sort of going to be the focus of my most of my talk here today. So these are nocturnal sweat bees. You can see a picture, beautiful picture from Kristen Ziegler here of them on a balsa tree flower. It's big and bright and up in the canopy and attracts nocturnal pollinators. So that's one of the fun facts about these bees, is that they're nocturnal. Really interesting part of their biology that I won't be talking about at all today. But I'm picturing them here over an overview picture of Barro, Colorado Island, which is in the middle of the Panama Canal. It's owned and protected for research by the Smithsonian Tropical Research Institute, which is where I go to study these bees. And the reason that I go all the way to Panama to study this particular species is because of the remarkable social plasticity that exists within this single population. So let me just give you a little walkthrough of their life cycle so you get a sense of that. Here we have these are twig nesting bees. So this is sort of what we're looking for when we're looking for bee nests. And this is also what a newly emerged female would be looking for when she wants to start her nest. So looking for sticks like this suspended in vine tangles above the forest floor. Once she finds a nest that seems adequate, the first thing she does is dig a tunnel right through the center of it and then build a little entrance collar around the entrance that's just about the size of her head so she can adequately protect it from intruders. If we were to open up the side of that stick, we see that she's lined that tunnel with brood cells. These are just little individual rooms for her individual baby bees. And she makes them out of chewed up sawdust. So she chews up the wood and then builds these nice little cells. You can even pop them out pretty easily. And if you open one up here, you're looking at a larvae sitting on a pollen provision. So these are mass provisioning bees. They go out and they collect nectar and pollen, pack it all into one of these brood cells, and then lay an egg on top of it and seal up that brood cell. So if you look right here, you can see the little caps on those brood cells. So there are some larvae or pupae or eggs inside of there developing. It's about a 35-day development period, at which time they emerge from their brood cell. And if it's a female that emerges, then she has a choice. She can stay in her nest as a worker. And if she does that, there are a number of really important differences between queens and workers. This eusocial relationship develops. And so over the past 10 to 15 years, my collaborators and I, specifically Adam Smith at George Washington University, he and I have worked closely together to characterize these differences between queens and workers. We find that queens are behaviorally dominant. They get fed by the workers through trophallaxis, that's mouth-to-mouth food exchange. This is actually really rare in adult bees. We don't see this. And so it's pretty cool that they do this, actually. The workers will feed the queens. The queens are mating and egg laying, but the workers are not. They don't mate, they don't lay eggs. Physiologically, this, it's like what you would expect based on those differences in behavior. Here are some queen ovaries. They're nice and plump. They're filled with mature eggs. Here, if you look at these little strings here, those are the worker ovaries. They're really tiny and undeveloped. The workers are smaller. And then the queens have higher titers of vitellogenin and juvenile hormone. These are, it's an egg yolk precursor protein and a hormone that's involved in oogenesis, but many other functions. So that's what you would expect, given that the queens are reproductive and the workers are not. There's also many hundreds to thousands of genes that are differentially expressed between the queens and the workers. So real differences there. This is truly a eusocial relationship. But what's really fascinating about these bees is that the female that emerges in a nest that's not her only option. She can stay and be a worker, but she could also disperse and start a nest of her own. So let me just walk you through what that looks like. So here we have a little flow chart and a little cartoon that's basically showing you the same thing. If a newly emerged bee, like here in white, decides to disperse, then she is more than likely to end up as a queen of a eusocial nest. That is, in her first brood of offspring, she produces both workers and males. But we find that about a third of these foundresses do something different. They stay solitary. And they do this by producing only male offspring in that first brood. Now, in the social Hymenoptera, the workers are exclusively female. So by producing only sons, they don't even have the option of having workers. They remain solitary. Now, you may be thinking maybe they haven't mated. In Hymenoptera, sex determination is based on whether the egg gets fertilized or not. These females who remain solitary have mated. They have live sperm in their sperm storage organ called the spermatheca. They just somehow are not using it. They're producing only males. Okay, so with this, this social flexibility within a population, we have both solitary and eusocial nests. I've been using the species as somewhat of a proxy for this transitional state in the loss of independent replication to transitioning to the origins of eusociality, trying to understand how this can evolve through natural selection. And so, like I said, for the purpose of this talk, we're going to sort of walk our way around these 4 questions of Tinbergen, trying to integrate across levels of analysis and timescales as we go. So hang on, here we go. Okay, so first we're going to start by integrating across levels of analysis at the current timescale. And we can ask, how does genotype map to phenotype, and how does phenotype map to fitness? Or in other words, can we understand how it works by understanding what it is for? So by integrating across these levels of analysis, can we get a better sense of how it evolves? And here's what I mean by that. So if we ask the question, what causes helping behavior? Typically, the approach is to think about this in terms of altruism, worker altruism. And this has been done within using Hamilton's rule, this very famous inequality where the benefits of helping are scaled by the relatedness of those that you help. As long as those outweigh the costs of giving up your own reproduction, then you can have this altruistic behavior evolve through natural selection based on indirect fitness benefits. But there's another way that we can think about this. There's another potential mechanism, and that is parental manipulation. And that is if not so much that the worker is being altruistic, but is instead being manipulated by one of their parents to stay and become a worker or a helper. And so in this case, we can still think about this within the context of kin selection and Hamilton's rule. And here we'd be testing the prediction that the benefits of having daughters as helpers is outweighing the costs of investing in non-reproductive or dead-end offspring, those workers who are not laying eggs of their own. And it's really important to keep in mind that these are not mutually exclusive. Both can be operating at the same time, and they're both entirely consistent with kin selection. The difference here is that in the worker altruism model, we have the trait bearer is the one receiving the fitness benefits. But in this parental manipulation model, the mechanism lies within the parent, the trait is expressed in the offspring, and the parent is the one receiving the fitness benefits. So by understanding this mechanism, we can better understand how it evolves. Okay, so really simple, right? We just plug in the numbers to this really simple inequality. I would argue that this turns out to be one of the most complex equations in evolutionary biology. It's pretty simple, pretty straightforward to go out and measure relatedness these days, but understanding those benefits and costs is really complicated. So, for example, how much benefit do workers provide? And how should this benefit be attributed, divided up between queens and workers in terms of their own inclusive fitness? And how much reproductive success do workers give up? In species that are obligately social, it's really impossible to know what they could have had if they were solitary, because it doesn't make sense from an ecological perspective. They've evolved to be always social. But facultatively social species can give us at least some estimates of what those costs and benefits may be, because we can directly compare the outcomes of solitary individuals and social individuals. So that's what we did. We used observation nests to track reproductive success of new foundresses. So here is just like a three-dimensional nest that we've taken apart. This was a stick. We took out the walls, and you can see all the little brood cells. You might be able to see a little bee peeking her face out of one of those brood cells. We've taken this and smashed it into a two-dimensional frame, basically. So we have a piece of balsa wood. We've dremeled out a tunnel for them, and we put newly merged females inside of there, seal it with a piece of plexiglass, and then hang it in the forest. So about half of these females, we do this, and about half of them just take off. We have no idea where they go. It's a total loss, but about half of them stick around, and then we can track their lifetime reproductive success. So we can check on them every 4 days, see when they've built cells, when they've closed cells, when new offspring emerge. We can measure, we can videotape their behavior inside the nest and get a really clear picture of what the consequences of social and solitary life are. Okay, one caveat here is that I said we seeded each of these nests with a newly emerged female, right? So one thing that we have not measured here is dispersal costs. So that could potentially be a huge factor in inclusive fitness that we just can't measure. And I'd be happy to talk about this during the Q&A with you. But based on what we could measure, here's what we find. The inclusive fitness benefits of working, of being a helper at the nest, are like really a bad deal. They do not outweigh the cost, the reproductive costs of giving up reproduction. So what I'm showing you here is our estimate of inclusive fitness for 180 foundresses and 80 workers. We had still, despite the fact that we could get really nice estimates of the benefits and costs by comparing solitary and social nests, we still had to make some assumptions in calculating inclusive fitness. And that's because we don't know how to divide up those any extra work offspring that are produced in social nests, because it really depends on who's in control, right? So we calculated inclusive fitness in two different ways. One, assuming that daughters are completely in control and they get all the benefit of extra help, or that mothers are completely in control and they get all the benefit? The real answer is probably somewhere in between, but this gives us, some extreme estimates. But either way you slice it, the inclusive fitness of foundresses is far greater than the inclusive fitness of workers. And what's really remarkable is that a third of our foundresses died before any of their offspring emerged. So their inclusive fitness was 0, and yet they still on average do better than workers. Okay, so what about the different social strategies of foundresses? Well, we could compare those two. And so again, either way you slice it, on average, whether you pursue a solitary strategy or a social strategy and become a queen, or even pursue a social strategy and fail to become a queen, it really, in general, works out the same. Your inclusive fitness is basically the same. So what this suggests to us is that in this species, there's not a ton of evidence that there's been selection for worker altruism, but there does seem to be support for the predictions of helping behavior coming through parental manipulation. Okay, so does understanding what it is for help us understand how it works? I would argue that worker behavior may be for helping moms to increase their inclusive fitness, and it therefore works via maternal manipulation. So we can sort of continue making our way around this, these sets of 4 questions, and now we can integrate across time scales at the proximate level of analysis. First, by asking, okay, well then how does development influence the mechanisms of behavior? And specifically, we can ask, how do mothers manipulate their daughters into working? What are they doing to that developmental process in the daughters that causes them to behave as workers? So we had to get at this in several indirect ways. It's actually really hard to manipulate these bees too much during development. So we collected circumstantial evidence. So if moms are manipulating their daughters into working, then we would expect that they are usually producing 2 types of female offspring, some that are going to be workers and some that are going to be dispersers. And if a potential mechanism for altering their developmental trajectory is nutrition during the larval stage, then we would expect to find more variance in the quality and quantity of those pollen provisions that they make for female offspring than they make for male offspring, because we assume there to be just sort of one optimal male type or son type, that the type that can go out and get as many mating opportunities as possible. So what we did is we collected the pollen provisions of eggs from natural nests. We genotyped the eggs to get the sex, and then we measured the dry weight and the protein content of those pollen provisions. And sure enough, we find that the variance in the pollen provisions given to female offspring are higher than those provided for male offspring. Now, what are the potential consequences of that? Well, here what I'm showing you are a couple measures that are representative of differences between queen and worker casts. So we have head width as a proxy for body size, and then we have ovary size as a proxy for reproductive potential. These light blue squares here are workers that we've collected from observation nests. You can see they tend to be small with small ovaries. And then the darker blue circles are dispersers. These are ones who have naturally colonized new nests, so they've recently dispersed. So they're about the same age as workers, but you can see that they're larger and have larger ovaries. Now, all these gray triangles in here are females that we reared out of natural nests. So they had eaten their pollen provisions provided to them by their mothers, but then we took them and reared them up in the lab for 10 days, where we gave them as much food as they wanted, but they didn't have any sort of ecological cues, any sort of social cues. And despite that, we see that some end up looking a lot like dispersers and some end up looking a lot like workers, suggesting that offspring reproductive potential may be influenced by maternal manipulation of larval nutrition. We also have reason to believe that mothers could be manipulating their daughters into helping by being aggressive towards them. So here we've tracked behavior inside those observation nests, and we did this in 2 types of nests. One that we called incipient nests. These are where the worker was less than 10 days old. She just emerged. She was just sort of like drying out her wings and sort of getting used to the world as an adult, and hadn't really started performing the duties of a worker yet. And then we had established nests, where the worker was fully engaged in worker behavior and was older than 10 days. So here we see trophallaxis, that mouth-to-mouth food sharing behavior. And incipient nests, the majority of this pie you can see is orange. That means that the direction of food sharing was from queen to worker primarily. But in established nests, that reverses and the worker begins feeding the queen. But look at aggression, it's different. We have in those first 10 days, those early days, the queen is beating up on her worker, and that doesn't change even once the worker starts working. She keeps being aggressive towards her. And this seems to have consequences. So here we experimentally manipulated the conditions that do happen in nature. We get these what we call replacement queens, where the queen can die or disappear, and one of the workers can take over as the queen. And so we just replicated this at an experimental level, where we left a single worker alone in the nest. We removed the queen and any of her siblings. And what we found is that she develops her ovaries and starts laying eggs at the same rate as those solitary females. So workers are capable of having high fertility. It's just that when the queen's around, they don't. We also looked at this at an endocrine level. This is just a schematic that shows some of the various functions that juvenile hormone has in insects. It's been related to migration, mating behavior, egg-laying behavior, and especially oogenesis, so the development of eggs and oocytes. And so we measured juvenile hormone in a bunch of different types of female Megalopta. Here you can see when we rear females in a cage in the lab, they don't really start producing juvenile hormone. But in our observation nests, you can see queens have relatively high levels of juvenile hormone. Those solitary females have lower levels. But here's what I want to draw your attention to. These sets of females are all about 10 days of age. Some of them are workers, some are new nest foundresses that we put in those observation nests and let them go for 10 days. And then some are natural dispersers. They've colonized natural nests. And so we think they're about 10 days of age. You can see there's drastic differences. So the workers, despite being the same age as these groups, have significantly reduced levels of hormone. So how do mothers manipulate their daughters into helping? Well, it's possible that females are manipulating their daughters' reproductive development and potential by suppressing those endocrine pathways via nutrition during the larval stage and aggression once they emerge. Sorry, I keep hitting the arrow. Okay, so still sticking with this integrating across timescales at this proximate level of analysis, we can ask, well, what are the mechanisms that create these different developmental environments? And so specifically, we can ask, what causes some mothers to manipulate their daughters? And what's really cool about Megalopta is that here we can take advantage of the fact that there are these foundresses who pursue these two different strategies, and we can make direct comparisons between the social foundresses and the solitary foundresses. And so when we do that, we find that these solitary females are really pretty worker-like compared to queens. They're smaller, they have smaller ovaries despite the fact that they're reproductive. They have lower levels of juvenile hormone. There also seem to be a lot of life history differences and trade-offs. So the solitary females, here's sort of a little timeline of their life cycle. The solitary females lay their first egg about 2 days later than the social females. That doesn't seem like a lot, 2 days, but it was a significant result. They do catch up. If they're going to have a second brood of offspring, they lay that starting on the same day on average. But what ends up happening is this trade-off between the number of clutches and the size of those clutches or broods, as we typically think about it in insects. And so here in blue, we have the solitary females, and in green, we have the social females. You can see in brood 1, they have the same number of offspring. But keep in mind that for those social females, a lot of those first brood offspring are reproductive dead ends. They're workers who will never reproduce. And then in later broods, the solitary females produce far fewer offspring, and really they're much less likely to produce any brood at all or any offspring in later broods. They're typically like one and done. They have a first brood of offspring and then that's it. Whereas the social females go on to produce additional offspring in later broods. So there seems to be this trade-off between early and continued reproductive success that really seems to work out to pretty evenly matched reproductive success. The average lifetime reproductive success in those first 3 broods was close to 4 offspring or reproductive offspring for both solitary and social females. And I used to think, oh, that's a nice tidy result and we'll just end it there. But my colleague, Callum Kingwell, who just finished his dissertation work studying these bees, had the patience to let these observation nests go as long as they possibly could. And what he found is that actually there are really crazy differences in lifespan between these solitary and social females. So the average lifespan of a solitary foundress is just 2/3 of that of queens. This translates to over 2 months. And the maximum lifespan of a queen is 7 months longer on average than that of a solitary female. So twice as long, their maximum lifespan. But what's really fascinating is that it seems to be the workers that make all the difference here. So if you remove workers from these social nests, the average lifespan is about the same as that of a solitary female. And then they're much less likely to produce additional brood past their first brood. So this is the subject actually of some new funding that we're going to be starting. And so I'd be happy to answer more questions about this in the Q&A if you're interested. Okay, so we also recently sequenced the genome of this species, and we did some population resequencing of the whole genome to try to understand whether there are genetic variants associated with this difference in social strategy. Here I'm showing you a Manhattan plot. On the y-axis is FST. It's just a measure of genetic differentiation between populations. Even though our social and solitary females come from the same population, for the purpose of this test, we treated them as if they were 2 different populations, just to see where we might find genetic differentiation in the genome. And then each of here you're looking along the genome, and these are actually a bunch of different individual dots. It's just that they're super condensed and there's 4.5 million of them, so they look like little columns. But each of these dots is a SNP, or a variable site in the genome. And the orange ones correspond to really genetically differentiated regions of the genome against an undifferentiated background. So most of the genome, there's like no difference between solitary and social foundresses, but at these 26 different sites, we see large differences between them. And then if we look in those different regions that are genetically differentiated, we find several SNPs that are in coding regions of genes that are highly divergent between social and solitary types. And so a lot of these genes are involved in reproduction and neurogenesis, things that you might expect to relate to differences in social behavior. But this is really preliminary. Like it needs a lot more work to really declare that there's any sort of genetic basis for these differences in social strategy. Okay, so what causes some mothers to manipulate their daughters? Well, there's differences in life history traits that may regulate maternal manipulation of offspring development, and this could have a genetic basis yet to be determined. Okay, so let's move on here and start integrating across levels of analysis at historic timescales. So here we're going to start thinking about the role of development in evolution. And we can ask, how does developmental plasticity evolve, and what is the role of development in evolution? So first we can ask, okay, how do sensitivities to developmental cues evolve? And to answer this question, I want to zoom out again and go back to the family Hellictidae. And we are going to turn our attention to this group here, the Nominae. And within this subfamily, we have the alkali bees, Nomia melanderi. These are native bees to the Western US. They're actually really important pollinators of alfalfa seed. And what's cool about them is that they have a lot of traits and behaviors that you'd expect to be sort of precursors to eusociality. And they live in really dense nesting aggregations, even though they're solitary. they have their own nest, but it's really easy to work with them because they're so densely aggregated. So one of my PhD students, Mel Hagedorn, set out to understand how social interactions can influence brain development in these solitary bees. So trying to understand how cues to the social environment may be interpreted in the brain and evolve. So what you're looking at here is a beautiful confocal image of a bee brain. These are the antennal lobes. So the antennae would be coming straight out of your screen. You're looking at the bee from above. These here on the outside are the optic lobes. And then up here, these little cup-shaped things are called the mushroom bodies. You can see the three-dimensional rendering of them down here. These are the sort of cognitive processing centers of the insect brain. They're involved in learning and memory. All kinds of really cool things happen in the mushroom bodies. So what we did was went out to southeastern Washington, where these bees are nesting in these huge aggregations, and we put these emergence traps on the ground so that we could catch these adults as they emerge from their winter diapause. They diapause as larvae, and then they emerge as adults in the summer, and we could catch them right away before they've had any sort of experience, bring them into the lab and rear them in cages. And when we did that, we reared them in the cage either alone or with a partner, a conspecific. And then we looked at what that did to their brains. And so here you can see that for the most part, there's not a whole lot of differentiation, whether they were reared solo as in white here or paired in the gray boxes. But we do see significant differences in the development of the lips. The lips are this region of the mushroom body calyx. They're the gray part here. I don't know if you can see that. These receive input from olfactory neurons. So, suggesting that by experiencing a social environment, interactions from a conspecific, this enlarges the part of the brain that's receiving olfactory input, suggesting that that there could be some sort of predisposition in solitary bees to be responsive to cues from the social environment. So how do sensitivities to developmental cues evolve? Well, the ancestors of social bees may have been pre-wired in some way to respond to cues from conspecifics. Okay, so next we can ask, what is the role of development in evolution? And so specifically, what is the role of developmental plasticity in social evolution? So I just want to stop and define developmental plasticity for a moment, because there are lots of definitions out there. This is the way I'm thinking of it. It's a phenotypic response to internal or external environmental cues. And this has really been one of the hallmarks, one of the most celebrated features of social insects. So here we have a major and a minor worker from the same colony. They developed from the same genome. You can see they look drastically different. They have different behaviors. And that all is a result of developmental processes. And what I've been really interested in is, well, developmental plasticity is an outcome of social evolution, but can it also be a driving force? May it also be a cause? And I've been using transcriptional profiles to test this hypothesis that social evolution can be driven by developmental plasticity. So let me sort of walk you through the way I've been doing this. So we've been looking at plasticity in gene expression during development and between social types. So here, this should be familiar to you now. This is just the social life cycle of Megaloptogenellus. Here, I'm showing you all these different contrasts in gene expression that we've looked at. So we have differences in gene expression between social types, the workers, the queens, the solitary females, and then those replacement queens. We did this, we have transcriptional profiles of these in the brain and in the abdomen. That's what these little symbols represent here. We also have transcriptional profiles of these during sex differentiation, so males and females of different stages, and then also during holometabolous development or metamorphosis, so eggs, larvae, pupae, and adults. And I just want to point out that this developmental plasticity is very ancient. Holometabolous development evolved 400 million years ago. haplodiploid sex determination about 300 million years ago. And comparatively, eusociality is actually a really recent phenomenon. So by comparing gene expression in these different contexts, we're able to understand how plasticity during development is related to plasticity among social types. Okay, so here is the first result when we start to do that. And there's a lot going on here, so let me just walk you through it. What we're looking at here is a correlation matrix of gene expression differences. So on both sides of this matrix, we have differences during sex determination. So males and females during the pupal stage or during the newly emerged females or males. We also have differences in life stage. So eggs versus larvae, eggs versus pupae, pupae versus larvae, et cetera. And then we have differences in gene expression between social types in two different tissues, the abdomens and the brains. And the size of the dots in each of these cells of the matrix represent the degree of correlation. So the larger the dot, the stronger the correlation in gene expression differences. So there's a few things that you expect here along this, this matrix, right? You have differences in gene expression between any two life stages also are correlated with differences in gene expression between other two life stages. Same with social types, right? So differences between queens and workers tend to be highly correlated to differences between, say, queens and solitary females. But what's really striking here is what I've sort of highlighted in these two rectangle boxes, that differences in gene expression between social types are highly correlated with differences in gene expression between the different life stages and between the different sexes. This is strongest in the abdomen, but we also see it in the brain. And so this suggests to us that genes that play an active role in development also seem to play a role in differences between social behaviors. We tested this hypothesis in another way as well. Here we measured, we used model selection methods to understand the best predictors of social plasticity in gene expression. So what I mean by that is that for every gene in the Megaloptogenellus genome, we calculated a plasticity index in several different contexts. So the plasticity index tells you basically how variable gene expression is between social types. And the developmental plasticity index tells you how variable gene expression is across stages of development or across sexes. And then we use these model selection tools, and we found the best predictors of social plasticity for every gene in the genome tend to be these factors. And what I'm showing you here are the coefficients, the estimated coefficients. So basically, how strong is the effect? And what you see is the largest bars for social plasticity in the abdomen and in the brains tend to be plasticity across sexes and plasticity in gene expression across life stages. Again, suggesting that developmental plasticity is one of the strongest predictors of social plasticity. Okay, so for developmental plasticity to play a role in social evolution, we really need to know which mechanisms are regulating plasticity and how it's acted upon by selection during social evolution. And so to do that, we have been investigating some of the mechanisms of developmental plasticity and doing that by looking in gene regulatory networks. So here I'm just showing you sort of a diagram of some of these factors that influence gene regulation during development. One of them is the microRNAs. We've studied these in a comparative context, but I don't have time to tell you about that now. So if you want to ask me about that during the Q&A, feel free. But what I am going to show you is our results based on looking at differences in transcription factors. So transcription factors are just proteins that bind to the DNA, in the nucleus, and then that affects the transcription of nearby protein coding genes. So affecting gene expression patterns, basically. And the way we've looked at this is in a comparative context. So here I'm showing you what we predicted across 10 species of bees that vary in social behavior. For homologous genes, so say gene A in each of these species, if social plasticity is evolving through differences in gene regulation, then what we would expect is something like this that's depicted in this cartoon. So we have two homologous genes, but in the social species, in the promoter region of that gene, we have many more opportunities for regulation by a particular transcription factor through its binding sites than in the solitary species. And in fact, that's what we found. We looked at 281 transcription factors. These are highly conserved all the way back to Drosophila. And we ran some correlation tests between the strength and frequency of binding sites in the promoter of homologous genes and how that strength of binding potential correlates with social complexity. And so here is a bar graph. Each of these bars is a different transcription factor. And then what you're looking at, the y-axis, is the number of significant correlations between binding strength and social complexity. The part of the bar that's above the x-axis, so positive correlations, are in blue, and the part of the bar that's below the x-axis, so negative correlations, are in red. And what you can see is for the vast majority of those transcription factors, most of the bar is above the x-axis, meaning that there were many more positive correlations between binding site gains and increasing social complexity, corresponding to this little cartoon here, this prediction, where there's more potential to regulate the transcription of that gene through transcription factor binding in the social species than in the solitary species. So we looked at this also in the Megaloptogenellus genome. So remember this Manhattan plot here where we're looking at genetic differentiation across the genome. We had 131 outlier SNPs, meaning that they were significantly differentiated between our social and our solitary types. Sorry, we had many more than that, but 131 of them were in promoters or introns, indicating that they could be playing a regulatory role. And of those SNPs in the promoter or intron regions, 32% of them, about a third of them, affect transcription factor binding. So here's what I mean by that. Here's a little diagram of a gene. This is just some random gene. We named it 05716. And you can see here are two different SNPs in that gene, and they're in an intron. And so the color of these dots represents a different SNP and corresponds to a different transcription factor. So for example, here we have the BAB1 transcription factor. In the social females, there tends to be an A at that SNP. And in the solitary females, there tends to be a C at that SNP. And this motif here shows you what this transcription factor looks for to bind to DNA. And here is that little star that shows you where that SNP is. And so you can see that this transcription factor prefers to bind where there's an A at this locus. And so it's more likely, it's significantly more likely to bind to this intron, to this particular intron in a social female than it is in a solitary female. So potentially these genetic differences between social and solitary females could be regulating gene expression. And we find that many of these transcription factors that are affected in this way are regulating neurogenesis or endocrine pathways, which we can think may have a role in social behavior. Okay, so what role does developmental plasticity play in social evolution? Well, it may be priming gene regulatory networks for social plasticity. Okay, so finally, and this one will be quick, I promise, we can think about integrating across timescales by asking ultimate level questions. So first, how does evolutionary history influence current utility? I don't have time to tell you about this today, but this is an area that we've just started doing some work in. So if you want to ask me about this in the Q&A, I'd be happy to share more about that. But we have been doing a little bit to ask about what is the current utility, how does that influence evolutionary trajectory? And we've been doing that by asking, how does social function shape molecular evolution? So if a gene is involved in social behavior, what does that mean for how it evolves at a molecular level? And so going back to the Megalopta genome, we used that social plasticity index again. So again, how variable gene expression is between the different types of social behaviors in both the abdomen and the brain. And we, again, used model selection methods to identify the best predictors of neutrality index. So the neutrality index measures the direction and degree of departure from neutral evolution. So if the neutrality index is 0, where this red dashed line is, then this is a signature of adaptive evolution. But if it's above 1, this is a signature of negative selection. And what we find is that for genes that have, so each of these dots is a gene here, and the color of the dot indicates how old the gene is. But we can see that for genes that have a really high degree of social plasticity, so that is there is a lot of differences in gene expression between the social types, they tend to have a neutrality index around 0, indicating that they're undergoing adaptive positive directional selection. It's actually the best predictor. Social plasticity index is the best predictor of neutrality index in both the abdomen and the brain. We measured this in a different way. One additional way, we looked at positive selection, genes undergoing positive selection as identified by a McDonald-Kreitman test. This identified 68 genes, and a quarter of those are differentially expressed between one or more social types in either the abdomen or the brain. This is, a relatively modest overlap, but it was statistically significant, indicating that genes with a social function are enriched for genes under positive selection, indicating that the function of a gene in its social behavior may be influencing its molecular evolution. But here's the kicker. We repeated that analysis for genes that have other modes of plasticity, specifically during sex differentiation or life stage or holometabolous development. And what we find here are these 17 genes that are here, right, that are both undergoing positive selection and also differentially expressed between one or more social types. Of those 17 genes, all but one of them is also differentially expressed between life stages or across the sexes, between males and females. So suggesting that selection on genes involved in social behavior is really influenced by their role in development or in developmental plasticity. Okay, so what effect does social function have on evolution? Well, social function may be shaping molecular evolution, but pleiotropy matters. The role that those genes are playing in other forms of plasticity, and specifically during development, may be having an even stronger effect on its molecular evolutionary trajectory. Okay, so just to wrap up here, reminding you that the big question was trying to understand how the loss of replication at the individual level can be selected, can evolve through natural selection given its fitness costs. When we synthesize across these 4 questions from Tinbergen, we can, start to find some clues here. So helping behavior may be the result of manipulation that benefits moms, at least in Megaloptogenellus. Moms who vary in life history strategies may manipulate their daughters through nutrition during the larval stage or aggression once they're adults. Social plasticity might emerge from tinkering of gene networks related to development, and the social and developmental plasticity may be shaping the evolutionary trajectory of those genes. And so with that, I just want to acknowledge a ton of collaborators without which this work would not have been possible. My outstanding group of graduate students and undergrads, and then funding. And I would be happy to take your questions. Thanks so much for your attention.
Thank you, Karen. Thanks for a great talk, and it's cool to see Tinbergen's 4 questions addressed in that way. Really fascinating. So at this point, we have time for questions. And again, just a reminder to people here, please post a question mark in the chat, and I'll relay you to Karen to answer the question. And if you're in YouTube. So at this point, please start dropping your question marks in the chat and we'll get the discussion going. And just one more reminder, please, when you ask your question, please tell us your name, where you're from. We want to try and build a community here, and that's a great way to do it. So, so our first question comes from Anandita.
Hi, hey Karen, very cool talk, very cool system indeed. I'm Anandita. I have been working with social insects for quite a while now. I work with ants and I'm based in the Rockefeller University right now. So Karen, I was wondering for the Hellictid bees, does the mother also manipulate the proportion of social to solitary bees in some ways? And I was also wondering, what is the utility of only producing males for the solitary females, you know? And I mean, what does these males do? Do they go on to mate with the females, which will be Foundresses in the next season? So but it's really cool that such a behavior exists. So.
Yeah, thanks. Thanks for your question. So yeah, to answer the first part, the females are, producing both workers and dispersers or gynes. And so they're definitely having, if what we think is true, that they are actually doing this through maternal manipulation, either through the larval provisions or aggression, then they're clearly having to adjust that strategy. Unfortunately, we only have circumstantial evidence for this at this point. It's really difficult to rear these bees at the larval stage in the lab. And so an ideal experiment would be to actually manipulate their provisions. And we've tried this countless times and it always fails. And so hopefully someday someone much more clever than me will come along and figure out how to do this. But for now, we're just relying on all of this circumstantial evidence coming at it from these indirect ways. In terms of the males, so one thing I didn't talk about was a recent paper that was led by my primary collaborator, Adam Smith, looking at sex ratio between solitary and social nests. And we accumulated all of our data from years of collecting these nests in the field. And we found that there's some evidence for this split sex ratio hypothesis in this species, where the solitary females, by producing more males, are sort of getting a fitness advantage by producing sort of the rare sex, because the social nests then are producing more females on average. And so you end up sort of getting, this is one hypothesis for how social evolution can evolve in the Hymenoptera, is through the split sex ratio, where the social nests are favored because they are specializing on females, and this can be incentive for workers to stick around and increase their inclusive fitness benefits by rearing sisters whom they're more closely related to than their brothers. And then meanwhile, you can have solitary females benefiting by producing males, because that then becomes the more rare sex, and so they gain fitness advantages that way. And so there's some evidence for that in this species. As far as what males do, I mean, there's so many limitations. This is such a cool species, but there's so many limitations to working with them. We have no idea what the males do. They stick around in our observation nest for, about 4 days, and then they disappear, and we don't know. We don't know where mating happens. We don't know when it happens. So we're assuming that solitary females, that there's, one optimum male that solitaries can really focus on making those males, but we just don't know enough about them. Hopefully someone more clever than me will be able to figure that out someday.
Thanks, very cool system, Karen.
Okay, so our next question is from Adriana.
Hi, thanks, Karen, for such a nice talk and a very interesting topic. I apologize because my camera is not really working today. I didn't have the extra one, so that's why I also have my camera off. But my name is Adriana Maldonado. I am from Universidad de Rosario in Colombia. And my question is, at some point in your talk, you were showing us differences between the solitary strategy versus the social strategy. And you mentioned that for the social strategy, basically the reproductive success depended on the workers. When you remove them, they basically had even lower reproductive success and similar lifespan than the solitary. Am I correct? Okay, so if that's the case, what's the mechanism that workers do? Like, what's the function? How do they make them have this increase in reproductive success?
Yeah, so the answer is we don't know yet. This is something that we're going to be like a new project that we're starting in the lab. But there's a lot of sort of initial evidence to suggest a few different things. So one is like really just basic, right? Like once the queen has workers, she gets to just kick back and stay in her nest. She rarely leaves. The workers take over the vast majority of the foraging. And so she's really capable then of just focusing on egg laying. All of her nutritional input, all of her energetic input can go into reproduction. And she doesn't have to, expend energy on flight or finding food. I mean, as I showed you, she's even getting fed by her workers. And so, that's like a really simple explanation. But in terms of like what the mechanism is there, whether it's that she just has reduced extrinsic mortality, she doesn't have to leave the nest, so she's less likely to get caught in a spider web or be eaten by a bird or whatever happens to them when they're out flying. It may also be that she's able to delay senescence because she doesn't have to expend metabolic energy on flight. She doesn't have an accumulation of free radicals and oxidative damage in her cells. And it may also be that there are some differences in the molecular sort of mechanisms by which reproduction and lifespan are occurring in the solitary and the social females. We know that there are all these differences physiologically and behaviorally in the social and the solitary foundresses, and there may also be some differences in the way they are able to sort of use energetic limitations. So this is a project that a grad student in my lab has started looking at trade-offs between immunity and energetic inputs and cast. And we see that like across all Megalopta females, there is a difference, like a decline in immune response with increased reproductive development. But for queens, this doesn't seem to be the case. They're able to maintain high levels of immunity despite the fact that they are, highly fecund and investing a lot in reproduction. And so it's possible that there is some sort of physiological shift that happens in queens and workers that allow them to continue to be reproductive when workers are around. So I guess the answer is we don't know, but we're looking into it soon, hopefully.
Fascinating, thank you.
Okay, our next question is from Jingyu.
Hello, I'm Jingyu, PhD student from University of Strasbourg and Cinerette. Sorry, my camera is not working well. So I have a question about the developmental plasticity and the social evolution. So in your opinion, why do you think these bees kept their developmental plasticity instead of becoming a full eusocial insect like many other bees?
Yeah, I mean, that is a great question. A few years ago, I would have said like, look, there's balancing selection. They have the social and the solitary females have exactly equal reproductive success over their lifetime, right? But as I showed you, subsequent work from Callum Kingwell showed like, oh, maybe that's not exactly true. If you let these things run longer, then you find that the social females are actually living a lot longer and probably producing a lot more than those solitary females. It's really tricky, you know, there's like, I try to be really careful about how I talk about and think about Megaloptogenellus. Like as I said, I use them as a proxy for that transitional state, but we don't really know, right? It may not be a transitional state. I mean, even just the question of whether the ancestor of Megalopta was solitary or social is a little bit open. For right now, it seems as though the ancestor was solitary and they have evolved eusociality from a solitary ancestor. But, you know, the phylogenies change all the time. There are a lot of things to be worked out there. So it's even possible that we're looking at some sort of loss of sociality, right? So I try to be really careful about the way I think about and talk about them as in a transition. We don't know that they're in a transition. This is, you know, this may just be the perfectly, you know, optimum behavioral lifestyle for this particular species. There's a second species of Megalopta on Barrow, Colorado Island, Megalopta amowina, that does basically the exact same thing. I don't talk about them as much because they're just a little bit less common. So we tend to focus on genalis instead. But you know, it's hard to even distinguish them by eye because they are so similar. So maybe this is just the optimal way to live in this particular environment. I don't have a great answer for it.
Yeah, okay, thank you.
Thank you for your question.
So we now have a question from YouTube. Tammy Ho writes, she's a PhD student from Aarhus University in Denmark. She says, thanks for a fantastic talk. How do you sex bees during development for transcriptome factor analysis?
Yeah, so that's why that's a great question. So like by eye, you can sex them as pupae because males have one additional segment on their antenna. And so especially when during the pupal stage, when the antennae are lying down flat, they're just clearly much longer than the females. So it's really obvious. You can sex them genetically with from the egg stage, even just based on genotype. So, you know, if you were using, for example, microsats or even like SNPs, you could just look for individuals that are homozygous at every locus would be a male because they only have one copy of their genome. And then individuals that are that have any sort of heterozygosity are females because they have two copies. So and then you can do some calculations to, you know, understand what the probability of your females being homozygous at every single locus are, but usually those are pretty small, especially with something like a transcriptome where you're using multiple, you know, millions of loci. It's pretty accurate. So that's a good question.
Okay, so our next question comes from Clara.
Hi, this is a great talk. Clara Jones, Clara B. Jones, mammals. I have two quick comments and then a question. Since the major transitions approach is a general model, even though one would not necessarily expect all of the transitions and components of those transitions to apply to vertebrates, many of the topics and questions that you raise could apply to vertebrates also. So since I often hear from vertebrate people that they don't understand how social insect literature could apply to vertebrates, just recall that the major transitions approach is general. The other thing quickly I wanted to say is that in the 70s, there was this big debate between Robert Trivers and Richard Alexander about whether or not offspring could ever, quote unquote, win. And your data, your findings about the importance of parental manipulation certainly probably applies to that debate. Now, finally, my question is related to dispersal costs. Isn't there a fairly straightforward way to measure that? And I haven't thought this through carefully, but what if you compared the mortality of dispersers with the mortality of non-dispersers? Couldn't you get a handle on that? And couldn't you even possibly study that in the lab?
Yeah, that's a great question, and I also appreciate the comments that you made before. I was telling Lauren before we started that, I started as a carnivore biologist, even though I was in a social insect lab, and I was heavily influenced by, when I was designing my PhD work, which was going to be on coatis originally, I was heavily influenced by the social insect literature. And even though I ended up switching to bees because for me, it was like, oh, there's no greater cost. There's no greater evolutionary cost than just completely giving up reproduction and being a sterile worker, and plus this species where it's like they can be both solitary and social in the same island is like, how can you pass that up? But anyway, so yeah, I appreciate those comments. And I agree wholeheartedly. But to answer your question about dispersal costs, this is definitely a major limitation for us. We have done some calculations to figure out like, okay, well, what would the dispersal costs need to be to make it be a better deal for the workers? And it's exorbitantly high. And, it's hard to believe that dispersal is as costly as it would need to be for it to be a good deal for workers. That said, we don't know, right? In our view, it's like we're out looking for these nests every day, and we find a zillion sticks that don't have nests in them that seem like they'd be perfectly good sticks for nests. So it's like, how can they be limited by sticks in a forest? There's like so many, but we don't know. That like we're not bees. So in terms of how we can get at it, yeah, you would think it would be pretty straightforward, but unfortunately, it's not. So once these bees disperse from the observation nest or even from a natural nest that we're tracking, we just have no idea where they go. We have never seen a marked bee. It's just too dense of a forest. Like I said, there's a zillion sticks. And so there doesn't seem to be any, it would be really lucky if there was some sort of phylopatry where they just disperse into a neighboring stick, and stick around, but that doesn't seem to be the case. And we've tried a little bit to do this in a lab, or at least some sort of like semi-artificial enclosure, like putting up like a big, like a screened in sort of bug dorm type of a thing, and putting flowers in there and things like that out in the forest, but it just doesn't work. They're pretty finicky. We haven't been able to get them to mate in the lab, for example. So it's pretty hard to know. But I would love to be able to fill that little gap in our, well, it's a big gap, but to be able to fill that. If anyone has any ideas about how we could do it, I'm all open, but they're not the easiest bees to work with. There's some things that are super easy and great about them, but some things that are a little bit challenging.
That's great. Thank you.
I have a couple of questions, but I'm going to remind people, if you have them, toss them into the chat and I'll prioritize those other than me. Mm-hmm. So I guess I'll just start. So Karen, you were talking about Yeah, so and please apologize if I forget things. I'm doing multiple things here, trying to track everything. So I might have missed it. The benefits in terms of the worker benefits of the females you were talking about, it seemed like that it could potentially enhance survival of the breeding females. So do you think this is a case of load lightning? Do the workers allow the females to forage more, to do other things?
Yeah, I mean, basically what it allows them to do is just be, just focus on egg laying. They don't really leave the nest very often after the workers emerge. So they're able to just offload, like you said, a lot of the energetic outputs and also, time intensive tasks. That said, like these are not egg laying machines, so we've all heard the stats about like honeybees, can lay like hundreds or thousands of eggs in a single day. I mean, these bees, as I showed you, their lifetime reproductive success was an average of 3.95. They're for especially for the tropics, they're really slow, and they'll often construct a brood cell and then provision it over several days, and then it'll sit open, and then maybe they'll lay an egg, and so it's like they're not laying eggs every day. There's several days in between when they lay eggs. It doesn't seem to have a ton of rhyme or reason when they're laying their eggs. Sometimes it's like one a day, boom, boom, boom. And sometimes, they'll have an open brood cell sitting there for 5 days before they lay an egg. Hellictid bees are known for their extremely large eggs. Given relative to their body size, their oocytes are very large. And so it is a significant energetic investment to make these oocytes. And so they may be egg limited more than they are resource limited. And so, by being able to stay in the nest and get fed by their workers and not having to go out and forage, it's possible that they're just saving a lot of energy and able to increase their reproductive output in that way.
Is egg mass related to survival and or different behaviors? And again, I'm sorry if I missed that.
No, that's a great question. I have absolutely no idea. There is some evidence for that in other species of bees, but we haven't looked at that at all. It's really hard. I guess we could probably do it on like if we took some good X-ray images and then, tracked development of that egg. But it would be hard to do X-rays of these nests out in the field. Yeah. So it's a little challenging to get at that. I also study leafcutter bees, which we regularly X-ray all the time. Like every other day, we'll X-ray their nests, and then we can get an exact measure of the provision size and the egg size and all of that. And so we haven't looked at the differences in egg size, but we know the provision size in that species makes a huge difference in terms of their behavior and their physiology and everything else. So that would be cool to try in Megalopta, but I don't know. I don't have a good answer about what role egg mass plays. Yeah.
And I don't see any questions, so I'm going to continue on. You showed a figure so there was a figure where you were looking at like the extent to which plasticity in the sex and plasticity in life history influenced success, I think. And you had two sides to it. And one of the sides, there was a lot of variation explained by within group.
Mm-hmm.
I don't recall you mentioning that. Do you explain that a bit?
Yeah. Do you want?
Yeah, maybe share the screen.
Yeah. So let's see. Let me get back to it, and then I'll share that screen. Yeah. So this was measuring. This was talking about.
You don't see it quite yet. Hang on a minute.
Sorry.
Yeah. And I think there was issues with your pointer, so
oh no.
maybe use your mouse.
Oh, I'm sorry about that.
No, no worries. It happens.
Okay. Let's see.
Yeah, that one. So on the right there.
Okay. How about if I do this, can you see a laser pointer?
Yep. Yeah, I see a pointer now.
Okay, yeah. So here, what we're measuring is the factors that best predict social plasticity in gene expression. So like we took every gene in the genome and asked how variable is expression between the different social types in the abdomen and in the brain. And then we did all these model selection methods to find the best model. And so these are the things that come out. It's all these differences in plasticity index. And then we have like orthogroup properties. These are like the age of the gene, the copy number variation of the gene, the duplicability, so how often it's duplicated over evolutionary time, the evolutionary rate of the gene, and then, yeah, within group expression, so the coefficient of variation, and then the expression level. And so yeah, in the abdomen and the brain, the strongest predictors tended to be some of the strongest predictors. We'll get to this in a minute, but some of the strongest predictors tended to be the degree to which the gene is differentially expressed during sex determination or during holopentabulous development. What's striking here is that here's the social plasticity index for the opposite tissue, right? So this is explaining the plasticity index among social types in the abdomen, and social plasticity in the brain is a relatively weak predictor, despite the fact that they're the same exact individuals. We just took the brain and the abdomen from the same individuals. These individuals that we measured sex plasticity and life stage plasticity in are completely different individuals. It's a completely different study, a different data set, but yet they're the strongest predictors. So in fact, they were 3 to 19 times as high as the next strongest predictor, which was orthogroup age, actually, where that's a negative coefficient. So like the youngest genes tended to have the highest plasticity index, social plasticity index. So, okay, so yes, so getting to the coefficient of variation or the within group expression. So here, yeah, we see these differences where within group expression also tells you something about how variable the expression is going to be across castes. And so what this basically tells us is that within the brains, the gene expression profiles of social types are really highly variable. And those genes that are involved in creating that variation within social types are also playing a large role in creating variation among social types. So basically, like, this indicates that there are differences in the way social plasticity develops in different tissues. And I think that just tells us even more that plasticity, is really important here, because it differs across tissues, even within the same individuals. Does that help?
Yeah, thank you. Awesome.
Yeah.
We have a question now from Tomislav. You're on mute, Tomislav. Tomislav, I'm sorry, we cannot hear you because you're on mute.
Yeah, now you can hear me, I hope.
Yeah, can you please start again?
Yeah, thank you for the great talk. I was actually, I was just trying to recollect some old ideas when I observed your slides. And when you showed all these SMP or single polymorphisms, and then I recollected the Gerald Edelman's morphoregulatory hypothesis about development and about the regulation of the development. That is this Topobiology book from 1988, Gerald Edelman. He is a Nobel Prize laureate. He's dead. But Gerald actually postulates three kinds of genes regulating development. And it is morphoregulatory genes, historegulatory genes, and selector genes. So I was wondering, and he says, of course, already in the 80s, he was emphasizing the importance of epigenetic control of development. So I know this is kind of a ex machina question right now, but it would be very interesting to know if you could pinpoint any of those genes that you have, how do you call it? You use it as a snapshot, or you are doing a survey of the genes. But is it any sign of a different regulation, which Gerald Edelman has called morphoregulatory or historegulatory or selector genes, because he was speaking already in the 80s about different genes coming up in a different moments in development. It would be interesting. I don't know if you have been thinking about in these terms. Thank you.
Yeah, no, thank you. I am unfamiliar with those terms, and so that would be really interesting to follow up.
Yeah,
on.
that is Topobiology. Gerald Edelman. The book is published. He was a Nobel laureate. Topobiology: An Introduction to Molecular Embryology.
Yeah.
And it would be interesting to see, because there are so many papers published when people are doing a snapshot of a genome, and then people are looking for a p less than 0.005.
Right.
But I mean, the point is, can we really pinpoint on a different regulatory functions of those genes that are deviating from 0.005? So that was my question, only.
Yeah.
Or maybe suggestion.
Yeah, so that's what we did with the transcription factor binding analysis, looking to see if some of these SNPs that are highly divergent between the social and the solitary types, if they could potentially be affecting the way transcription factors bind to the promoter regions of these genes and therefore influence regulation. We also have done some comparative work with microRNAs, which also play a regulatory role in development, turning on and off different mRNA transcripts by sort of targeting them for degradation. And we have looked across 6 species of bees actually to characterize all the different microRNAs in the genome and then identify what their potential targets are, and then look to see whether those targets have been of like any new genes in new species are targeted by, any new microRNAs are targeting genes that are involved in social behavior. And we find some evidence for that. We also have been looking at microRNAs in the context of development in some work that I've been doing with a solitary species. So looking sort of at maternal, not quite maternal manipulation, but maternal effects, like how the mom's condition influences what she's packing into her oocytes. in terms of messenger RNAs and microRNAs and what effect that has on offspring development. We find that does differ. So if moms are skinny and sort of undernourished and don't have a lot of lipid stores, then they tend to put different types of messenger RNA and microRNAs into the oocyte.
I think microRNAs are especially interesting in the context of epigenetic control. I mean, that is out of question. And again, thank you. It was just a suggestion from my side, nothing more.
Yeah,
Thank
sure.
you.
Thank you. I'll definitely check that out and look more at those 3 types of genes you're talking about. That sounds really fascinating.
Thank you.
Thank you.
Sorry, I was muted. Eduardo, please go ahead.
Thank you, Karen, for a very interesting talk. And I apologize, some of you may have noticed I needed to take a phone call regarding some doctor's appointments. So I apologize if my question or my asking has already been addressed. And I'll let you choose. I mean, we're getting to the point where just a few of us, so I usually like these last minutes because we can go places. So I apologize if you've already talked about the circadian biology of the bees, but you mentioned, I didn't even know that they're nocturnal bees. And I'd like to hear some more about what's been done or what you have done regarding understanding the evolution of nocturnality and diurnality in the bees.
Sorry.
You choose. And the other one is, for those of us working with systems where we cannot even dream about having some kind of proper sociogenetic approach to what we do, when I try to the best of my ability to follow everything you've done, sometimes it just goes over my head, I have to admit. I want to invite you to imagine, share with us your thoughts. Where do you see the state of the field 10 or 20 years from now? I mean, some 20 years ago, when you were probably not even thinking about bees yet, we had a whole genome sequence done. And I always tell the story that those of us studying behavior felt like, oh, that's it. I mean, they're going to have the sequence of bases and bingo, no need to study phenotype. It didn't happen. And the genome-wide association studies didn't really produce as much as we were told they would. Now we've moved from the sequences to the expression, and we're having these humongous amounts of data relating things. Can you just, for the sake of having fun, imagine if you were to bet some money on where we're going to be with sociogenetics 10 or 20 years from now, where would that be? Your choice of what you want to talk about.
Well, I'll answer both because the answer to the first question is very short. I have done nothing to understand the evolution of nocturnality in bees. It's really fascinating. I have some really talented colleagues who have specialized in this. For example, Eric Warrant at the University of Lund in Sweden has been studying the physiology of nocturnal vision in Megalopta and a lot of other insects. And what they find is that Megalopta does not have a nocturnal eye, like what you would expect in like a moth or something like that. Instead, it has a typical diurnal eye, but is much better at integrating light. And so they're actually light limited. And that's actually something I should have mentioned before when I was talking about how these are kind of lazy bees and don't really produce many eggs. They're also probably like food limited because they only forage for about an hour before sunrise and an hour after sunset. And often that's actually more like 20 minutes before sunrise and after sunset. So it's a really limited foraging window for these bees. And there's also been, my colleague Simon Tierney did a large phylogenetic analysis of the evolution of nocturnality in bees. It's actually evolved several times in bees. It's really fascinating, but that's all I know about it. One idea is that it's to avoid competition with diurnal bees. There could also be some physiological, like, thermal limitations there, which is an area that I've a colleague, Victor Gonzalez, has begun studying, and I've been sort of engaging with him on that, and he has some really cool ideas about that. So anyway, that's all I know about that, but it's really fascinating. In terms of sociogenomics, I think I am certainly not a visionary in this area. I will tell you that I think it's becoming more and more easy to access high quality genomes. What used to be a major project is now just sort of almost, it's pretty well standardized, and we can generate like chromosomal level assemblies now for bees, which have relatively small genomes in, a matter of weeks. There's currently a large initiative to sequence, the Earth, what is it called? The Earth Genome Project. I'm butchering the name. I can't, for some reason it's escaping me right now, but the goal is to sequence every, genus on Earth. And so I think it's only a matter of time before you will also have a high quality genome for your study of, your study species of interest, and be able to do really nice things with it. One thing I'll say is that, when we're still in this phase of like having pretty good genomes, but not quite completely closed genomes yet. And that limits what we can look at, especially going back to the earlier question by Tomislav, like when we have decent genomes, it's really easier to look at things at the gene level, because it's easy to align orthologous genes across species and identify genes. They have certain features and characteristics that make them relatively easy to identify. Regulatory sequence, all that stuff in between genes, that's much more difficult to identify if your assembly is not perfect. It's also more difficult to do comparative analyses with, because they're more difficult to align. Like conserved genes are pretty easy to identify and align, but all that regulatory non-coding sequence it's much more difficult. And so to do evolutionary analyses or comparative analyses is more challenging. However, there are new methods being developed all the time, and now it is possible to do that. So I think that, pretty soon, like we'll be able to look at regulatory sequences in a much more detailed way, a much more informative way, and get a better sense. Because like you said, like when you started, it's like, oh yeah, we're going to find the gene for altruism, like that's how, like I did my PhD in behavioral ecology. That's what all the models are. You start with a solitary population. There's an allele for altruism that invades. Can it spread through the population? Well, now we know there's no, we knew that, but the phenotypic gambit has become even more like obvious now that we know that there's no single allele for altruism. It's really complex, and there's probably a lot of regulatory alleles that are playing a role. And so being able to harness that, and do comparative analyses on that is still a great challenge. But I think the time is coming.
but if I can follow up on this, and this, you mentioned the phenotypic gambit, because maybe I wasn't clear on my question. That's exactly what I was wondering to hear from you. The problem is that there is no phenotypic gambit anymore. It's just a purely genetic gambit. And so, but what I feel is that again and again, because that's where the money is, they keep telling us that the answer is going to be in genetics. 20 years ago, it was the GWAs, and now the moment you start scratching the surface, we learn about all these associations they find, which tend to explain 0.5 of the variation. And we're losing, my concern as a behaviorist is that there's no similar focus and emphasis in understanding the phenotypes. Even if we get all the specifics of the regulatory genes, where are we going to be in 20 years from now? Because they're going to have some new kind of genetic approach to this. And we will still be lacking the behavioral data, the phenotypic data to make sense of that. That's where, but again, that's my probably very naive, almost ignorant view as a behaviorist. And I worry, since you are bridging more the two, are we going to find 20 years from now that all these associations that we're doing, okay, now we have to try again because we don't really cannot predict how the animal is going to behave.
Yeah. So I think that's an excellent point, and something that I've thought a lot about too, is that, okay, well, now it's getting easier and easier to generate genomes, but like, unless you know something about the natural history of the species that you have the genome for, it's, what are you going to do with that in terms of trying to understand the genetic basis for behavior? And so, generating the natural history information has become much more difficult than generating the genomic data. And so I think that that's someplace where we're going to have to make a very deliberate effort to stay on track and be learning about the natural history and the behavior just as much as we're learning about the natural history of the genome. As far as like linking GWAS studies to behavior, I think that functional genomics is an area for ripe development. GWAS studies provide a hypothesis that can then be tested, right? But unless you have the tools to test that, it's very difficult. And there, NSF has this EDGE program for developing genomic tools in functional genomics in non-model species. And so there's been a ton of advancement in that with like wasps and ants. And I think that, that also is coming along, but it's certainly still a challenge for non-model organisms to, take the results of your GWAS or some sort of association or correlation study and then take the next step to understand what role it really has in behavior. I think that's definitely a challenge.
I want to hope that it's going to be colleagues like you. I mean, because the power is going to be in genetics. The power meaning the money, that's where the journals are publishing, that's where the field is going, and I don't think we can fight that. We need more people like you who are going to be doing the genetics, but with an understanding that my story is going to be incomplete. I'm not going to make the progress I want unless I have the natural history, unless I have the behavior. I'm having maybe a somewhat pessimistic view of how behavioral ecology can really move the dial on its own. It'll require that we hear from the geneticists saying, I'm going to be doing this grant for functional genomics, but it's a waste of our money unless we have a behavioral component going with the grant as well. So hopefully you'll be leading the way. Thank you so
well,
much.
I don't know about that, but if I review that grant, I will give it high marks.
All right, so at this point, I'm gonna stop the live stream, and then people just want to chat. That's great. I know Clara had a question for you. So Clara, just hold on until I stop the live stream.
Sure.
And so for everyone who's still here, thanks for being here. And for those on YouTube, thank you. And
Thank you.
Karen, really appreciate you, your great seminar.